Welcome back, biology rock stars. You guys are the people who have pushed through, have mastered displaying how different traits are inherited in a variety of different organisms. You've mastered monohybrid crosses with simple dominance or complete dominance inheritance. You've looked at multiple alleles and codominance with blood typing. You've looked at incomplete dominance with palomino horses. You've also looked at sex-linked traits such as maybe night blindness, Duchenne's muscular dystrophy, Fragile X syndrome, hemophilia, color blindness, things like that. You've looked at pedigrees, and now you're ready for the ultimate challenge of dihybrid genetics problems. These are my favorites. So again, welcome. Here's a problem that we're going to look at. Here it is. A heterozygous tall, heterozygous round pea plant is crossed with a short pea plant that has wrinkled seeds. What fraction of the F1s will be tall and round? Make a key, determine the parents' genotypes and gametes, perform the cross, and provide a phenotypic ratio of the F1s. Wow, there's a lot to do. Again, what makes these dihybrid crosses different from the other kinds of crosses that you have done in the past is that you're looking at the inheritance of two traits at the exact same time, how information is passed down for two different genes at the exact same time. So we've got, in this scenario, we're looking at the height of the plant, and we're also looking at the texture of the seeds that plant has. So whether it's tall or short and whether it's round or wrinkled. Now, there's a lot of information that I gave you in the question stem, and there's a lot of clues that are in the question stem that help you to figure out what's dominant and what's recessive. If at all possible, I'm always going to give you those clues. And I'm going to expect you to use those clues to figure out then what the dominant phenotype is, and what the dominant allele is, and what the and what the recessive allele would be. So the fact that we have these organisms that are heterozygous, and that we have a phenotype connected to that heterozygosness, that phenotype would be the dominant one. Another clue that we have in this question stem to know maybe what's recessive is that I just tell you that these plants are being crossed with a short pea plant that has wrinkled seeds. And I didn't tell you if that short pea plant is homozygous or heterozygous. Because I didn't tell you that pea plant is homozygous or heterozygous, I'm in fact giving you the recessive phenotype, and you understand genetics, so you know that the genotype of this organism has to be homozygous recessive, and the same for this particular trait. The organism would be homozygous recessive in order to have wrinkled seeds. Okay, let's set up our key, because that's the first thing listed in our tasks. Make a key and then determine the parents' genotypes and gametes. So our key, I'm using T because T is the first letter of the word tall, and that's the dominant phenotype. I'm also going to use the letter R because the other dominant phenotype is round in regard to that other trait that's being inherited at the exact same time. So there's big T for tall. There's a little t. It should come up. There we go. For short, a big R for round, and a little r for wrinkled. Pretty simple key. Okay. Parents' genotypes are next. My key is done. Determine the parents' genotypes. So the parents, let's see. The first one is heterozygous tall, heterozygous round. So to be heterozygous tall, your genotype would be big T, little t. To be heterozygous round, your genotype would be big R, little r. So the first parent's genotype is big T, big R, little r. The second parent is short and has wrinkled seeds. So its genotype would be little t, little r, little r. Again, we're looking at the inheritance of two traits at the exact same time. These pea plants are diploid. They have pairs of chromosomes. This parent pea plant has one chromosome that has this recipe, in a sense, big T on it. The other chromosome making up this pair would have a different recipe on it, resulting in the wrinkled texture. Oh, I'm sorry. It's actually short. It's not the wrinkled texture. In regard to the other pair of chromosomes that are present inside of this parent's, all of the cells of this parent, one member of that pair of chromosomes would have a big R recipe on it, and the other member of that pair would have a little r recipe on it. So the parent genotypes are big T, big R, little r, being crossed by the other parent, the one that is wrinkled and short, little t, little r, little r. Okay, so next in our list is to come up with the gametes. Now the gametes, okay, this kind of a Punnett square, this kind of a cross, it's just bigger than what we've looked at so far. We're going to end up making a 16-box Punnett square, a 4x4 square. And in order to figure out what are the possible combinations for the gametes, you kind of do a little bit of like the FOIL method in math. We see what are the possible combinations that a pea plant could have in regard to genotype for the height as well as for the texture of the seed. This particular pea plant could produce a gamete that has a big T in it and a big R. This parent plant could produce a gamete that has a big T in it and a little r in that gamete. This parent plant could produce a gamete that has a little t and a big R genotype. And the last possible genotype for a gamete for this first parent here that's heterozygous tall, heterozygous round, it could produce a gamete that has recessive alleles in it. It could be little t, little r. So there are the 4 possible gametes that the first parent could make. Big T, big R, big T, little r, little t, big R, and little t, little r. Now the other parent is homozygous for both traits, so there's not going to be a lot of variation amongst its gametes that it makes. It only has certain recipes. It can't create a big T recipe. It doesn't have it. So the gametes that this particular parent, the second parent, is going to make will have the genotype little t, little r. A second gamete would result from the combination possibly of this first little t and that second little r. A third gamete combination would be looking at the second T in the genotype for that short pea plant, this little t combining with the first little r in the genotype. And the fourth one, the fourth genetic combination for the gamete would be a little t, a little r, the last of those in the genotypes. There they are. Not very exciting, not very different from each other, but that's probably true for a lot of the different genes that we have too. We don't have any variation inside of our cells. I like having a 4-chambered heart. Okay, let's set up our Punnett square. I told you it was going to be a 4x4 or a 16-box Punnett square. They're really big, so that's why I gave you a lot of room on your note sheet. Probably the most critical thing here is that you make sure that you put your gametes in the right places. You don't mix up at this particular point in time, mom's gametes and dad's gametes. So all of the gametes that are produced by the first parent go on one side of the Punnett square. The gametes that are produced by the other parent go on the other side of the Punnett square. So we end up with a possible gamete, here it is, from the first parent, big T, big R. A second possible gamete, big T, little r, little t, big R for the third, and little t, little r for the fourth. The other parent's gametes will put on the left side of the Punnett square. There they are. Now when we go to do the cross, because that's what we're supposed to do next, we have our gametes, then they're in the right place, perform the cross using our Punnett square. We're going to bring our gametes, the alleles down and over from the gametes. But we're going to keep T's together and we're going to keep R's together, just like what we did when we set up the genotypes for the parents, our heterozygous tall, heterozygous round. So when these two gametes basically join their chromosomes together or donate their chromosomes, add their chromosomes to each other, we'd end up with an offspring that was heterozygous, big T, little t for height, and also heterozygous for the texture of the seed round, a big R, little r. And we see it there in that first box of the Punnett square. Here's our big T, little t, our big R, little r. If by chance fertilization were to happen between the second gamete that the first parent produced and the first gamete that that second parent produced, we'd end up with a genotype for that offspring of big T, little r, little r. And that's what's listed here. Again, keeping the T's together and the R's together. Here's our third result, little t, big R, little r. And again, there's our fourth. If this gamete were to be fertilized by this gamete, we'd end up with an offspring that was homozygous recessive for both of the traits, little t, little r, little r. Here are the rest of the results. Again, bringing alleles down and bringing alleles over, we get all of these possible results for our F1s. These are F1s, or first filial generation. They are the offspring of these parents, or the parental cross. Okay, the next thing that we're supposed to do is we're supposed to list out the phenotypes so that we can list out a phenotypic ratio. So what are the potential phenotypes that pea plants could have in regard to these two traits? We could have tall pea plants and round. We could have tall and wrinkled. We could have short and round and short and wrinkled. And that's what I've got here. One potential phenotype would be a tall, round offspring. Another potential phenotype, just because we've got this particular key here, we could have tall and wrinkled offspring. Looking at the other phenotype for height, we could have a short, round offspring. There it is. We could also have a short, wrinkled offspring. And there we go. So those are phenotypes. The potential phenotypes from these two parents, for any parents, I guess, in regard to, for any parent, the potential phenotypes for the offspring in regard to any height cross combined with the texture cross would be tall round, tall wrinkled, short round, and short wrinkled. So now we have to look back at our Punnett score and we have to figure out what are the actual phenotypes for these offspring. Well, I noticed something here. I noticed that we've got all of these gametes here that are genetically identical to each other. That's one thing that I noticed. I noticed something else here, that we have all of these offspring are genetically identical to each other in this column. These offspring here are genetically identical. These offspring are genetically identical. Those are genetically identical. It might make it easier when we have to count up how many we have of each phenotype. So the genotype big T, big R, little r would result in a phenotype of tall round. And so we have 4 tall round. Big T, little r, that would be tall and wrinkled. So we have 4 offspring that would be tall wrinkled. Little t, big R is the genotype that would result in short round offspring. So we have 4 potential offspring that would be short and round. And then short and wrinkled, we have 4 as well. So those are our phenotypes. 4 of the offspring in this cross were tall and round. 4 of them were tall and wrinkled. 4 of the offspring were short and round, and 4 of them were short and wrinkled. So because I have my phenotypes listed in this order, we would say that the phenotypic ratio is 4 to 4 to 4 to 4, which can be simplified to a 1 to 1 to 1 to 1. Again, the phenotypic ratios tell geneticists a lot about the parents. For example, there could be a 6 to 2 to 6 to 2 phenotypic ratio. Geneticists often talk about a 9 to 3 to 3 to 1 phenotypic ratio. It could be 16 to 0 to 0 to 0 for a phenotypic ratio. And again, that would tell geneticists a lot about the parents. We could have a 0 to 0 to 0 to 16 phenotypic ratio, where all the offspring are homozygous recessive for both traits. We would know for sure then that the parents are also homozygous recessive. A lot of information can be inferred regarding the parents when you have a phenotypic ratio. Okay, here's your challenge problem. Oh, I forgot. Always have to go back and make sure that I've done what I'm supposed to do. What fraction of the F1s will be tall and round? 1/4. Tall and round. We have 4 out of 16. That's 1/4. Okay, now here's our challenge problem. I'm going to set up the problem with you. Here it is. And then you guys are going to work on it on your own, and you're going to show me what you've got when you come to class. And then, only then, will I let you do the practice problems, which are really fun. Okay, cross 2 heterozygous tall, heterozygous yellow-seeded pea plants. Make a key, determine the parents' genotypes and gametes, perform the cross using a Punnett square, and determine the phenotypes of the F1s and their phenotypic ratio. Okay, so 2 heterozygous tall, heterozygous yellow-seeded pea plants. All right, you guys ready? Okay, go for it. As always, let me know if you have questions. Let me know what you need from me. Have a great day.